Okay, hello guys. So, last class, I know that we finished all the slides in the mitosis notes. So now we're on to meiosis. And the first part of meiosis was talking about asexual versus sexual reproduction, and we finished those as well. So, before we can talk about mitosis specifically today, we have to talk about chromosomes and what they look like in different organisms, including humans. So, you all know by now that we need to be filling in the blanks in your guided notes. I'm going to go a little bit slower in this video than the other one because I know I go way too fast. Sorry, guys. But do not panic. I'm also going to post these. This video will be posted so that you can watch it and fill in the missing blanks. All right, so let's talk about chromosomes. So by this point, you all know human somatic cells have 23 pairs of chromosomes. Why? Because humans specifically have 46 chromosomes total. We also already talked about somatic cells. Somatic cells are every cell in your body except the gametes. Somatic cells, remember, have 2 chromosomes, one from mom, one from dad. All right, so in karyotyping, I think some of you probably have heard this before or seen it. Karyotyping is the idea that we can order the display of the pairs of chromosomes from a cell, and we order them, from largest to smallest, and that's the easiest way to organize the chromosomes. So pair 1 is going to be the longest pairings of chromosomes, one from mom, one from dad, in this big white box. As we organize them and continue to pair them up, we go from bigger to smaller. So by the time we get to the end, we have really tiny chromosomes, and those are the last pairings. Karyotyping is important in other species because we have cataloged ours, right? But other species are going to be different because they have different numbers of chromosomes and they have different lengths and sizes of chromosomes. If there are genetic disorders, this is where we can also identify, hey, maybe they have an extra chromosome. Maybe they don't have a certain chromosome. So we can get data from this type of information as well. You can see that the 2 chromosomes in each pair, usually they are the same size because they are called homologous chromosomes or homologs. If something's homologous, that means the same. So in this case, homologous pairings are going to be exactly the same because there's one from mom, one from dad. The chromosomes in homologous pair are the same length and shape because they carry genes controlling the same inherited characteristics. So if on chromosome 1, for example, let's say it's gastron 1, he's this pink part right at the top. It's the same on your mom version of the allele and your dad's version of the allele. So that's why we call them homologous chromosomes. All right, so the sex chromosomes are different. We got to talk about these separately. They are going to determine the sex of an individual, and they are called X and Y. Human females usually have 2 X's, and then human males have an X and a Y. All the remaining pairs of chromosomes are called autosomes. They have a special name. We call those autosomes. These are the sex chromosomes. And you can see that the Y is a lot smaller than an X is. This is why I was trying to tell you guys that a Y chromosome only has around 100 to 200 genes on it, but an X chromosome could have up to 900 different genes on it. So the size of a chromosome does determine how many genes are on it. Okay, so before we go any further, this is also something that I struggled with when I was learning genetics. I just want to make this really clear. If you understand haploid versus diploid, this is going to save you a lot of stress about trying to understand what's going on in meiosis and down the line when we talk about plant regeneration and stuff. So, haploid versus diploid. They play a role for determining how many pairs of chromosomes there are, but each pair of homologous chromosomes includes 1 chromosome from each parent normally, right? The 46 chromosomes in a human somatic cell are 2 sets of 23, one from mother, one from dad. A diploid cell is going to have 2 copies of each chromosome. We call it 2N. N is usually a chromosome. If there's 2, that means diploid. Di means 2. The stem di means 2. Hap means half. So that means you have 1 instead of 2. For humans, the diploid number is 46 because we should have 2 of each type of chromosome. Because 1 from mom, 1 from dad, that means we have a total of 46 because we're going to have 2 pairs. Now, haploid, let's just say we have 1 chromosome from a parent, not both parents. The haploid number would be 23. So we'll talk more about haploid in a second. Okay, so a gamete, a sperm or an egg cell, remember they only have one chromosome, one set, because mom has one, so the egg cell has one. That's haploid. They have half as many chromosomes than normal, so it has a single chromosome. A sperm cell, same thing, has one single chromosome. That is known as haploid. We call it just N, not 2N, it's just N. So for humans, the haploid number is 23. Each set of 23 has 22 autosomes and a single sex chromosome. In an unfertilized egg, the sex chromosome is X. In a sperm cell, the sex chromosome could be either X or Y. It depends. Okay, so hopefully that helps you see the difference between haploid and diploid. It's just counting the number of chromosomes present in a current cell. Okay, so we do have to talk a little bit more about chromosome sets in the human life cycle. So we already talked about fertilization. Fertilization happens when the gametes are unionized, so they come together. The fertilized egg is called a zygote. This zygote has one set of chromosomes from each parent once it's fertilized, because the sperm will give one, the egg will give another. So now you have two. And the zygote will then start producing somatic cells by mitosis and develop into adult. So that's why when I showed you the salamander video, you had one single cell that divided into hundreds to thousands of cells to help make a baby salamander. Yes, that's by mitosis. As soon as that egg is fertilized, we are in mitosis. So at sexual maturity, the ovaries and testes are going to produce haploid gametes. The gametes are the only types of human cells produced by meiosis rather than mitosis. Meiosis results in one set of chromosomes in each gamete. So an egg cell should have one, and a sperm should have one. Fertilization and meiosis alternate sometimes in sexual life cycles to help maintain the chromosome number. Sometimes they need to be haploid, sometimes they need to be diploid. Depends on the life cycle and what phase they're in of the cycle. All right. So again, gametes are the only haploid cells in animals. All the other cells are going to be diploid because they're going to have more than one chromosome. They're going to have two in there, one from mom, one from dad. They are produced by meiosis, and they're not going to undergo any other cell division before fertilization. Meiosis happens first. Gametes are going to fuse to form a diploid zygote that divides by mitosis to develop into a multicellular organism. So first we have meiosis, and then we go into mitosis as soon as we're developing a multicellular organism. This is just a picture to help you see like the cycle. So there is a cycle. Meiosis is going to occur, and you're going to get a little N. N means haploid, okay? So during meiosis, you have gametes that are haploid, right? As soon as these two gametes come together, one N plus another N is going to equal 2, right? So when fertilization happens, you have one N plus another N, and now you have 2N, and that's your zygote. As soon as that 2N is formed, now we're doing mitosis. And then you get a multicellular organism, like this little mouse. And then the whole process starts over. That mouse has a form of a haploid cell, whether it's a sperm or an egg. It's going to combine through fertilization, and then boom, you have 2N again, meaning you have mitosis occurring. Okay, so plants are weird, okay? Plants have a different form of this. There's kind of similar names, but it's called the alternation of generations. They do mitosis, but they also do meiosis, but they use spores. So they are going to do alternation of generations, which plants and some algae do this. This life cycle is going to include a diploid and a haploid stage, just like we have in humans. So you can see how this yellow on the top is going to be the haploid stage. The little n means haploid. The green will be 2n, meaning it's going to be the diploid stage. Okay, so the diploid organism called the sporophyte, so that's this green mass thing here. Sporophyte is considered diploid, and he's going to make spores. And those spores are going to end up being haploid spores. And those are generated by meiosis, so there's variation. Once those spores are out, they're going to go find the gametophyte. The gametophyte and the spores will join together through fusion to make a zygote, and you get mitosis. Sometimes the gametophytes are actually formed from spores through mitosis. All right, there's another slide with a little bit more information on this coming soon. Sorry about my voice, guys. I know it's terrible. Okay, so each spore grows by mitosis in a haploid organism called a gametophyte. So the spore is going to start growing by mitosis, and it turns into the gametophyte. The gametophyte then makes gametes by mitosis. Fertilization will happen of the gametes. You'll have a zygote, and that zygote through mitosis will turn into a sporophyte again. And then it's a cycle, okay? So this cycle will continue. As long as you have sporophytes, you're going to generate spores through meiosis. The spores will grow by mitosis into a gametophyte, and the gametophyte are going to then make gametes. They're going to fuse together to make a zygote, and then the whole process starts over again. This is the alternation of generations. This is how most plants will do this. Okay, so here's a picture. It's a little more complicated because we're talking about spores now. Okay, so if we start with a diploid multicellular organism, here's a plant. That's the sporophyte. Some plants and algae are this way. They start out as diploid. Through meiosis, they generate spores. The spores are now N, meaning haploid. They only contain one chromosome. Through mitosis, these spores are going to grow a little bit, and then we're going to create the gametophyte. The gametophyte will then undergo mitosis again and fertilization, and then it'll make a zygote. As soon as it's a zygote and fertilization has occurred, it is now a diploid organism again, where it can undergo mitosis to grow until it's a full-grown plant, and then the whole process starts over. That plant creates spores by meiosis. The spores can then grow a little bit, undergo mitosis to do that, turn into a gametophyte, which then creates gametes. The gametes will then join together through fertilization and make a zygote, and the process starts over. So hopefully this is a little easier for you to see the haploid and the diploid and what's going on. The fungi and protists. I'm not really going to go into this a lot. I just put this slide in here to remind me to tell you. Fungi and protists, which are single-celled organisms, sometimes they're a little different, okay? Their process is not alternation of generations, and it's not exactly fertilization like in humans. They undergo meiosis. They then grow a little bit. They form gametes. They do fertilization. So those guys are a little bit weird sometimes. Okay, so depending on the type of life cycle, either haploid or diploid cells can divide by mitosis. It depends on where they're at in the cycle. However, only diploid cells can undergo meiosis. So 2N, only diploid 2N cells can do meiosis. In all the three life cycles we talked about, the halving and doubling of chromosomes is what's going to help us get genetic variation in offspring. This is one of the keys for genetic variation. Okay, so that's the first introduction to do haploid and diploid, but we need to talk a little bit more about how meiosis occurs, right? So meiosis is going to reduce the number of chromosome sets from diploid to haploid, meaning it's going to go from 2 chromosomes from each parent to 1 to haploid, okay? So, like mitosis, meiosis is preceded by the duplication of chromosomes. So, the first step of meiosis, we got to copy those chromosomes. Meiosis is going to take place in two sets of cell division. So, unlike mitosis, where it's just one round and it's done, and you get two new cells, we're going to undergo two sets of cell division. One is called meiosis 1, and the other is called meiosis 2. So, two phases of cell divisions. The two cell divisions are going to give us 4 daughter cells rather than 2 daughter cells in mitosis. So, we're going to end up with 4. However, each daughter cell has half as many chromosomes as the parent cell does now. It's going to have one of each set of chromosomes instead of 2. Okay, so let's talk about the stages of meiosis. So for a single pair of chromosomes, homologous chromosomes, in a diploid cell, both members of the pairs are going to be duplicated. So we got to copy the chromosomes, right? This gives us sister chromatids. We already talked about those. They get stuck together, right? And they're closely associated because they have certain lengths, right? Homologs may have different versions of genes called an allele. So we've already kind of talked about an allele. The correct definition of an allele is that there's one of two or more DNA sequences occur at a particular gene locus. So there's DNA sequences that happen at a particular gene. And those genes could be an overlapping of DNA. So, for example, if you look at this picture, you have a little b and a big B. Those are two versions of the same gene. Your mom has one version, your dad has another. This is also kind of talking about like possibly the dominant genes, for example. We might get into that when we get into Mendel's idea. We'll talk more about alleles when we get there. Homologs are not usually associated in any obvious way except for during meiosis. It's the only time we really care about the homologs and how they pair up. Okay, so here's a big picture of what's going to happen. So when we start meiosis, we're going to have a pair of homologs in the diploid parent cell. Okay, so one from mom, one from dad. Those duplicated homologous chromosomes are now going to be a set of 2 because we duplicated them, and now they're considered sister chromatids. In meiosis 1, we're going to separate those homologous chromosomes so that maybe we'll call the blue ones from our dad get put into one, and then maybe the ones we get from our mom are red, those get put in another. And then in meiosis 2, we're going to separate the sister chromatids, and then you have one of each side in a new cell, resulting in 4 new cells. Okay, this is where we're going to kind of go with this. Okay, so in interphase, we have a pair of homologous chromosomes in the diploid parent cell. The first step we need to do is we need to duplicate the chromosomes. We duplicate the chromosomes, and once we do that, we now have sister chromatids. A diploid cell with duplicated chromosomes is now what we have. This occurs usually in interphase, right? We copy the DNA in interphase. Okay, so when we start meiosis 1, the homologous chromosomes will separate so that all of dads are in one, all of moms are in the other. The haploid cells within those duplicated chromosomes are then going to separate into sister chromatids that are separate now. So we've broken the sister chromatids, and they're each in their own little cell. That's what happens in meiosis 2. So the goal of meiosis, you're going to have the total number of chromosomes very specifically. It's going to reduce the number of sets from 2 to 1, with each daughter cell receiving 1 set of chromosomes. In the first meiotic division, homologous pairs of chromosomes are going to pair up and then separate. In the second division, those sister chromatids that we had in meiosis 1 are each going to separate into their own new cell, meaning you now get 4 new haploid cells. That's the result of meiosis. Just going to wait a few seconds to make sure you guys all get it in there. Okay. So here's the big picture of what it all looks like, okay? Meiosis 1 and meiosis 2. You see some familiar pieces in here, right? So we're still going to do prophase, metaphase, anaphase, and telophase, and cytokinesis. You're just going to do it twice. So in meiosis 1, you're going to have prophase 1. Some of the same exact things that happen in each phase that we learned about in mitosis are going to occur in each of these phases. That's not different. We just have to figure out what's happening with the chromosomes more specifically, because there are some times where the chromosomes are going to have like crossing over or certain assortments that help give more genetic variability too as these are occurring. So we start with prophase 1. We're going to go into metaphase, and then anaphase, and then telophase and cytokinesis, which is going to result in 2 new cells, right? Those 2 new cells are then going to start over. We're going to have prophase 2. They call it 2 when it's the second phase, right? So prophase 2, we're going to have metaphase 2, anaphase 2, and telophase 2, and cytokinesis, which is going to end up resulting in 4. Through this process of meiosis 2, you're going to have separate sister chromatids. Through the process of meiosis 1, you will have separate homologous chromosomes. So we're just building a little bit more off of mitosis, and the steps are a little bit different. So as long as you understand mitosis, this is going to be easy, promise. Okay, so let's look more closely at what happens. Here's meiosis 1. So in meiosis 1, we're going to undergo prophase 1 first, right? So we have centrosomes and spindles that are starting to form. The nuclear envelope is going to break down, right? The chromosomes, well, actually they're called sister chromatids now, right? The sister chromatids are present, and they might undergo a certain thing called crossing over here. The fragments of the nuclear envelope disappear, and we go on to metaphase. Those chromosomes, remember, are attached to the spindle fibers here at the kinetochore. That's going to help align all of those chromatids right along the imaginary metaphase plate to help pull them to the middle. When we get to anaphase, those spindles are attached to the kinetochores, right? So that means they can now be pulled to the opposite side. You see how these chromosomes have little different colors on the end? Like there's a little bit of red here, there's a little bit of red here, there's a little bit of blue here. There is genetic variation here now because parts of these chromosomes have swapped with different ones. That's called crossing over. We'll talk about that too. In telophase and cytokinesis, we're going to have now 2 cells. We still get a cleavage furrow. Exact same things occur. And we have 2 separate cells in the end. In meiosis 1, this is just a closer up picture. If you need to see these pictures up close, you can just go through these slides more if you want to. In meiosis 2, so here's the big picture of meiosis 2. Our goal for meiosis 2 is to separate the sister chromatids so that we have 1 in each new cell. So prophase 2, we have the similar things happening in prophase. The spindle fibers are starting to spread out. The nuclear envelope is breaking down. If we go to metaphase, we line them all up along the middle. In anaphase, we start to pull them to either side. And then in telophase 2 and cytokinesis, we have 2 whole new cells forming with the correct chromosomes inside, and the nuclear envelope is going to come back, and the centrioles will disappear. And this will result in 4 new cells. And those are going to be haploid daughter cells instead of diploid. Okay, so let's break these down a little bit. So it's a lot more complicated than I gave off in those pictures, right? Prophase 1 is actually broken down into 5 other steps, okay? I'm not going to ask you to memorize these, okay? This is developmental, devo bio stuff, and it's just really complicated. But you do need to know when synapsis or when crossing over occurs and what phase. So in prophase 1, all those chromosomes that we had are going to start to condense so that you can start to see them, right? The homologous chromosomes are also going to pair up here. So they should align with their correct size and be together. We are then going to start the process of crossing over. See how in step 2 here, there's no name for it officially, but these chromosomes are next to each other, so they're just going to swap some genetic information. Crossing over occurs in prophase 1 of meiosis 1, okay? So here's a better picture of a complex that forms after crossing over occurs. As you get closer through meiosis 1 at the end of prophase 1, look, the nuclear envelope is starting to fragment and fall apart. So, in crossing over, the non-sister chromatids are going to exchange DNA segments. So not the sister chromatid, not the copy is what's separating. It's the ones that just happen to be next to them. This is important because crossing over helps us get more genetic and variability. This is a good thing. Very specifically, so it does have a name where crossing over occurs. I'm sorry, I forgot. They call it the Pacatine stage. So the Pacatine stage during prophase 1 of meiosis 1 is where crossing over occurs. If you, for our purposes, say it happens in prophase 1 of meiosis 1, that's good enough for me, okay? Each homologous pair has one or more X-shaped region. We call these chiasmata. Chiasmata are then going to exist wherever there's a point that crossing over has occurred. So chiasmata only are called this after crossing over has occurred. And it's like a little X-shaped region. Hang on. Okay, so you need to know what chiasmata are and what crossing over is and where it occurs. Okay, so let's talk about metaphase 1. In metaphase 1, those homologous pairs are going to line up at the metaphase plate with one chromosome facing each pole. The microtubules from one pole are attached to the kinetochore, just like we talked about. The kinetochores are attaching to the spindles, the microtubule spindles. And that's what occurs in metaphase 1. Nothing fancy. We already know about that, right? Nothing new here. All right, so in anaphase 1, we're going to split up the pairs of homologous chromosomes. We're going to separate them. One chromosome is going to move towards each pole, and we have this happen by the spindles. The sister chromatids remain attached at the centromere, though, here, so they do not separate the sister chromatids yet. We're just pulling one of those down. And the centromere helps them move towards the pole. So this is the difference. In anaphase 1, the sister chromatids aren't what separate. We're just going to split up those chromatids onto different sides. All right, telophase 1 and cytokinesis. So in the beginning of telophase 1, each half of that cell now has a haploid set of chromosomes. Each chromosome is still a set of chromatids, though. So we still have things stuck together. Cytokinesis, remember, it begins in the start of anaphase, but it's still going on through telophase 1. It's going to simultaneously form 2 haploid daughter cells because the cleavage for row will form and split. And just remember, in plants, it's not a cleavage furrow, it's a cell plate. Now, a key difference here too is that there is no chromosome duplication between the end of meiosis 1 and the beginning of meiosis 2 because we already did it. We already replicated all the chromosomes that we needed to do before we started meiosis 1. So we don't need to do it again for meiosis 2. It's already been done. Okay, so hopefully you guys are thinking this isn't that bad. This is almost the same as mitosis. We're just adding a few things in and a few more steps. I'm going to wait a few more minutes, just make sure you're good before moving on. Okay, So the division of meiosis 2 also happens in 4 phases. So we're going to have prophase 2, metaphase 2, anaphase 2, and telophase 2, and also cytokinesis. Meiosis 2 is still, again, very similar to mitosis now. Okay? Remember, we still have sister chromatids stuck together at this point. So we're going to separate those sister chromatids now in this round of phases. So for phase 2, we're going to have new spindles form around the chromosomes. In metaphase 2, those spindles are going to help move the chromatids to the middle of that cell, just like we do in metaphase. They line up at the equator. The spindle fibers are attached at the kinetochores so that when anaphase 2 happens, now we separate those sister chromatids and we're going to have them pull to either direction of the cell. Then we start to get a nuclear envelope that forms around those new chromosomes, and the cytoplasm will divide. All right, so let's break it down. Prophase 2. In prophase 2, a spindle apparatus is going to form. In late, so there's different phases, guys. So there's early or late. So in late prophase 2, this means it just happens later, chromosomes are each still composed of the 2 chromatids. Those all line up towards the metaphase plate towards the end of prophase 2. In metaphase 2, the sister chromatids are arranged at the metaphase plate just like normal. And because of crossing over in meiosis 1, the 2 sister chromatids of each chromosome, they're not the same anymore. They are not identical. If you see this picture here, normally these are the same in mitosis. But because of crossing over, we have a little bit of blue here, a little bit of blue on this side. This whole one's blue. There's a little bit of red on the ends here. So they are not the same anymore. So the kinetochores are going to then start pulling those apart when we get to anaphase. All right, so in anaphase 2, the sister chromatids separate. Finally, they're separate. And the sister chromatids of each chromosome are now going to move into 2 newly individual chromosome poles. And then we get telophase and cytokinesis happening again. The nuclei are going to form, and the chromosomes start to decondense. At the end of meiosis, we should get 4 daughter cells, each with a haploid set of unduplicated chromosomes. So 4 new daughter cells that are haploid, and they are genetically distinct from each other. They are not identical because we did crossing over, right? So we now have different types of chromosomes in each set. Okay, so in telophase 2 and cytokinesis, once it's over, once again, you get 4 new daughter cells that are completely different from each other because we had crossing over occur, and this gives us genetic variability. All right, so let's go over a comparison of mitosis and meiosis. All right, so mitosis is going to conserve the number of chromosome sets, producing cells that are usually genetically identical from the parent cell. So they're the same. And you should have a diploid version because you should have 2 sets of chromosomes in those cells. Meiosis is going to reduce the number of chromosome sets from a diploid to a haploid, producing cells that are genetically different from each other and the parent cell. Meiosis typically undergoes 2 divisions after replication, each with specific stages. All right, so there are actually 3 events that are special to meiosis, and all 3 occur in meiosis 1. One is synapsis and crossing over. So synapsis is a version of like pre-crossing over stages. You can kind of include it with crossing over. This occurs in prophase 1. We talked about this already. The homologous chromosomes, they physically connect and exchange parts of genetic information with each other, just who's ever close to them. That's what they do with crossing over. When there's alignment of the homologous pairs at the metaphase plate, these pairs of chromosomes are positioned randomly on that plate, okay? So which direction they get pulled and which cell they get pulled in, that's up to the spindle fibers, and it's completely random. So this is another way that we get uniqueness in our cells for meiosis. There's also separation of the homologs during anaphase 1. Which cell they get pulled in depends on where they're located, right? And because every single one is genetically different, where they get pulled is what's going to happen with that cell. And which one cell has it, it's up to randomness. It randomly occurs. So these 3 events are also unique because synapsis occurs, that doesn't happen in mitosis. Alignment of homologous pairs at the metaphase plate. So we don't normally have pairs at the metaphase plate. We have a chromosome or a sister chromosome that's going to be turned into a chromosome at the metaphase plate. We are separating pairs. Also, separation of homologs during anaphase 1. We, again, are separating not just a single chromosome, we have a pair that we are trying to divide. So if you look at this picture for mitosis versus meiosis, the pairs is what I was talking about. That's different, okay? So in mitosis, we have duplicated DNA chromosomes. They line up on the plate like metaphase and then result in 2 N daughter cells. If we look at meiosis, during prophase of meiosis 1, you're going to have crossing over occurring. That's unique. That doesn't normally happen in mitosis. You're going to look at metaphase 1. So instead of having just, see how there's like red, blue, and there are 2 pairs right here? We're going to have a red and a blue right next to each other, and those pairs will get separated into pairs, not individual chromosomes like in mitosis. Those homologs will also separate in anaphase so that we have pairs that have been separated in anaphase 1. When we get to meiosis 2, then we separate those pairs into individual N haploid versions of the daughter cells instead of diploid versions of daughter cells in mitosis. So 2 versus 4. Okay, so these are important differences. You have to know the differences, okay? So at the end of this presentation, you guys are going to grab a piece of paper, and you are going to list the key differences between mitosis and meiosis. You can use your notes to do it, but you need to know the key differences between the two because that's going to help you remember what happens in meiosis, all right? And on a test, you're probably going to see the differences listed, okay? We're going to skip that because we already went over it. This is just closer up pictures if you want to look at it more closely. Okay, almost done. So the genetic variation produced in the sexual life cycles is what contributes to evolution, guys. So which genes get passed on from generation to generation contributes to evolution, and these changes, like mutations in the DNA, are the original source of genetic diversity. Mutations are good. We've already talked about mutations, right? That is what pushes us to have new abilities because we have new genes that do new things. Mutations create different versions of genes, which we now call alleles. So we can have different versions of a gene if it's slightly mutated. Reshuffling those alleles during sexual reproduction is what also gives us genetic variation through meiosis, which we just talked about. There are other ways that we can get genetic variation among offspring. So the behavior of chromosomes during meiosis and fertilization is actually responsible for most of the variation, but there are 3 mechanisms that are correlated: independent assortment of chromosomes, crossing over, and random fertilization. These 3 also contribute to randomness and genetic variability. I'm going to wait a second while you write those down. We're just going to talk about each of those, and then we're done. And you've survived. Okay, so what is independent assortment? So normally, homologous pairs of chromosomes are going to orient randomly at metaphase 1 of meiosis. So like I told you, those pairings that line up, they line up randomly. There is no order. In independent assortment, each pair of chromosomes are going to sort into maternal and paternal homologs, and those are going to separate into daughter cells independently of other pairs. So the chromosomes being sorted are not going together. They're randomly getting sorted into different positions, independently of sticking to anything else. The number of combinations possible when chromosomes assort independently into gametes is 2 to the power n, where n is the haploid number. So for humans, if our haploid number is 23, we know that, right? We have 23 pairs, 23 from mom, 23 from dad. If that's our haploid number, and we use this equation, 2 to the raised power of n, 2 to the power of 23, there are more than 8 million possible combinations of chromosomes here. Because independent assortment will assort these chromosomes independently of each other. They're not attached to each other, they're on their own. 8 million possible combinations. This is why we also look completely different from each other, which is a good thing, right? Genetic variability. Okay, so in crossing over, we've already kind of talked about it. You're going to produce recombinant chromosomes, which are going to combine DNA inherited from each parent. In meiosis, in humans, usually there's 1 to 3 crossover events per chromosome pair. That's actually kind of a lot if you think about it. 1 to 3 crossover events per chromosome pair. Chromosomes and their crossing over ability is going to contribute to genetic variation by combining DNA, producing chromosomes with new combinations of a maternal and paternal allele. So it gives us new combinations and genetic variability. Each zygote then has a unique genetic identity, so every zygote is different. Random fertilization. So in random fertilization is random. This also adds to genetic variation because any sperm can fuse with any ovum. The fusion of 2 gametes. There's actually technically 8.4 million possible chromosome combinations from independent assortment. As we just talked about, we're going to have a zygote with any of about 70 trillion diploid combinations. Yeah, that's a lot, guys. So there is a wide range of genetic variability because of meiosis and all these other parts. Sorry about the bell. I'm going to wait a second. Make sure you got it. Okay, so the evolutionary significance of genetic variation within these populations. What is the evolutionary significance? Natural selection is going to result in the accumulation of genetic variations that are favored by the environment. So remember, natural selection, those that can survive best in their environment are going to survive. And then they're going to pass on their genes to their offspring. And if they survive, it just keeps getting passed down from generation to generation. And these are usually favorable genetic variations, right? If they have an unfavorable variation, usually they don't live and they don't pass on their genes. Sexual reproduction also contributes to the genetic variation in a population. This is where population genetics comes into play. And a lot of people study population genetics, and they usually study like mutation sources and how that mutation occurred and how that genetic variation affects all the organisms around it. Usually asexual reproduction, like budding, we talked about budding, right, in the Hydra, those are really a lot less expensive than sexual reproduction is. Sexual reproduction has to go through mitosis phases, meiosis 1, meiosis 2. It's a lot more costly to a cell to do those things. But sexual reproduction is nearly universal among animals. This process of fertilization and the meiosis 1 and meiosis 2, nearly identical among all living things, okay? Overall, having genetic variability is evolutionarily advantageous. This is how we also get new species and biodiversity. Okay, I think that's it, guys. The last slide is just a summary of the 3 different types that you can look at if you want to all together. You don't have to. So be good. If you didn't get to this entire slide during one class period, do not panic. We are going to continue this when I come back, okay? So do not panic. This is just simply also a video. If you want to go over stuff again or listen to it one more time, because I know meiosis looks scary, you can look at this video anytime you want to, okay? All right, bye.